Okay, so one of the largest questions in this area of research is why is there such incredible variation in the penetrance and severity of FASDs? Why is it that some women who drink do not have affected kids while other ones do? And I think that if we take on aggregate the data that has been collected to this point, it's becoming very clear that a woman's alcohol exposure and her genetics alone do not explain the breadth of this condition, and so that perhaps, like in other areas of toxicology, male exposures are beginning to matter. And so this is kind of the perspective that I began with, trying to understand how it is the memory of an exposure could persist across generations. And so my interest in this area really stems or tries to understand at how the memory of paternal exposures is going to persist and then interact with maternal exposures to influence the penetrance of birth defects as well as chronic disease. And so for the relevance to FASDs, what I'm really trying to ask here is how is it that a father's drinking could potentially interact with maternal alcohol exposures? And so largely what I want to understand here is there an additive interaction where we have a 1 + 1 = 2? Is there a synergistic interaction? Or is it perhaps more nuanced than this, and that some phenotypes yes, some phenotypes no? And so as Sierra just introduced very eloquently, I switched my lab away from using a single parent model to this one right here, where we have a 2x2 factorial design where we examine the offspring of control. So when neither parent drank, we want to understand what happens certainly when dad drinks, when mom drinks, but then more often than not, we're really looking to this group right here to try and understand if there's any synergistic interactions between these parental exposures. And so to this end, we have a mouse model that Sierra has already introduced here, and so I'll just kind of recap this very, very quickly. And that is to say that we take the males and we're going to expose them to alcohol for about 6 to 10 weeks. And so in a mouse, this encapsulates 2 complete spermatogenic cycles. So it means that the sperm that they have in their reproductive tract have been exposed to alcohol throughout the entire genesis of that particular gamete or this particular sperm. And again, this is a C57 Black 6 mouse model. We use the drinking in the dark exposure paradigm, where we take the water bottle out of the home cage. We put in an alcohol bottle, a 10% alcohol bottle. We leave it in there for 4 hours, and in doing this, the mice generate these binge-like drinking habits. And then again, with respect to the females, what we do is we start the exposures 10 days before conception. We allow them to continue to drink until we can reliably diagnose pregnancy on gestational day 10, and then we pull the dams off alcohol, allow them to go through withdrawal, and then leave them alone until they either we harvest the tissues for analysis or they give birth. And so the blood alcohol levels that we see in this model are kind of around 90 to 100 milligrams per deciliter. And so just to put this into context, this is right around the legal limit or just slightly above. And so these exposure levels here are not pushing the envelope by any stretch of the imagination. And just to kind of emphasize the clinical relevance here, I stole this figure from a book by Tom Cook, who's really interested in the economic impacts of alcohol exposure. And he demonstrates right here that the top 20% of consumers are going to drink somewhere between 2 and 10 drinks a day. And so just to put this in perspective here, this largely encapsulates about 8.3 or almost 10% of adults of reproductive age. And so it's picking off a fairly large swath of society. All right. And so I want to put the rest of my talk in context here. I want to talk very, very quickly about some of the fetal work that we've done, just to kind of lay the stage here so we can understand, going forward what we're speaking of. And so in this data that I'm showing you right here, we're looking at offspring normalized birth weights. And so what we do is we weigh the fetus, we'll excise the brain, weigh the brain, and then take an equation here where we divide the brain weight by the body weight, and then we plot that against how much the parents drank. And when we do this, we see, just like in maternal models, we see these dose-dependent declines in offspring brain weights. And so this would be kind of similar to the clinical measures of microcephaly. And again, it doesn't really seem to matter at this particular outcome here, whether it's mom, dad, or both parents that drink. We see a pretty strong interaction, dose-dependent interaction on this phenotype. Okay, so one of the questions that we started with next was, how is it that this memory is transmitting from dad to offspring? But more importantly for our talk today, we really need to understand how is it that this biological memory of this alcohol exposure is persisting in the offspring. And so when we went back to our analysis of the male reproductive tract, and so this is the epididymis right here, when we went to embryos that were conceived using alcohol-exposed sperm, or when we went and looked at the placentas of offspring conceived by alcohol-exposed dads, what we consistently saw is a signature of mitochondrial dysfunction. So something is happening to the mitochondria, is either a primary consequence of alcohol exposure directly, or is in some memory of this exposure that's at least persisting on into fetal development. Okay, and so mitochondria are these tiny little organelles that live in our cells. They have these really cool proton pumps on the inner membranes that are going to take the molecules that you derive from your food, and they're going to use those molecules to generate energy. And so they're effectively like the generator or the battery of our cell. And so the hypothesis that we came up with was that we think that the exposure to alcohol by the parents, either preconception or early in life, in the case of the offspring, is in some way, shape, or form imparting some adaptive response to the mitochondria that's altering the thermostat in which way it functions, okay? And so mitochondria are these incredibly interesting and highly adaptable organelles that have allowed our species to survive and adapt to a myriad of different conditions. So there are humans that live in the Arctic Circle, there are humans that live in the desert. We have been able to adapt to all of these different environments. We have figured out how to adapt to a myriad of different food sources and get through different plagues, viral attacks, et cetera, et cetera. And so it makes evolutionary sense that parents are able to transmit signals to their offspring to modulate their mitochondria. And so what we think that we're observing here with respect to these long-term effects on offspring outcomes as a result of alcohol is some kind of maladaptive evolutionary signal. And this data that we've gathered to this point this far really suggests that it's this one component of the mitochondria, mitochondrial complex 1 right here, that is primarily affected. And so the data that support this are largely derived by work by other people that have been studying alcohol exposure directly, so not fetal alcohol syndrome. This is more the alcohol use disorder group, as well as studies that have studied or examined ischemic stroke, or accelerated aging. And I'm going to come back to that point here in just a little bit. All right, so what we wanted to ask in our model was, can we see evidence of this mitochondrial effect persisting in the offspring? All right, so to do this, we focused in again on complex 1, and we did an analysis that effectively tested the ability of complex 1 to generate power. And so I'm simplifying things here, but the analogy holds. And so again, we focused in here on complex 1, which is the larger battery component, if you will, of this particular protein complex. And the data that I'm going to show you very quickly is, of course, what we observed during fetal development. But the part that I want to start emphasizing more here are the long-term effects. And so we started to look in adolescence at a point that's kind of equivalent to, a mouse turning into their 20s. All right, so when we took our voltmeter, or we measured mitochondrial complex 1 activity, and what I'm going to show you here is the data for the male offspring of alcohol-exposed dads. And when we look at the brain tissue from these animals, what we see compared to the controls is that they have a reduced capacity to generate power compared to the control population. And more importantly, when we transition out of fetal life and then examine a mouse at a kind of equivalent point in life, whether it be in their 20s, and here I'm looking in their liver, what we see is a continued reduction in this, the ability of these mitochondria to function appropriately. Okay, and so as I said, mitochondria are these really wonderful organelles that have allowed us to adapt and thrive in a myriad of different environments. But when you have mitochondrial dysfunction, the biological system doesn't really know quite how to respond to that. And so it can tell something is wrong, but the only move that it's actually able to implement is an inflammatory response. And so this is specifically driven through the NF-kappa B and C-gas sting pathways. And essentially what happens here is that you start to get this low-level inflammatory effect that starts to creep forward, and it has these downstream effects that then lead into other conditions like cardiovascular disease, gut dysbiosis, metabolic syndrome, and then certainly aspects of cognitive dysfunction. And so we next wanted to ask, can we see evidence of this increased inflammatory state persisting across the life course? Not something that's just showing up during fetal development, but does this memory persist and cause this inflammatory phenotype long after or continuing into adult life? And so when we look at postnatal day 60, which is somewhere around the late teens, early 20s, what we see here is we do not see a signal if mom drank or if dad drank, but if both parents drink, we can absolutely detect an increase in inflammatory cytokines. And so the one I'm looking at here is IL-6. However, when we go forward into 6 months of age, and so this would be right around the time the mouse was in its equivalent to being in its 30s, what we see is we get a strong signal from mom, a strong signal from dad, but here again we have this additive interaction where the signal if both parents drank is much higher than what we see for either parent individually. However, by the time we get into postnatal day 300, which is kind of equivalent to being in your 50s, this inflammatory signal has kind of maxed out. And so this notion of inflammation and the relationship to FASD phenotypes is certainly not new. Joanne Weinberg has been speaking of this for many years, as well as several others. But the angle that is somewhat new here is the relationship to mitochondrial dysfunction. And so when we saw this, this really kind of prompted us to ask, could this be inflamma-aging? And so this word here is basically just a fancy fusion between inflammation and aging, but essentially what it refers to is this kind of low-grade chronic sterile inflammatory response that starts to creep up in our 30s, it increases a little bit more in our 50s, and then kind of gets to a high point here in our 60s and beyond. And really what this does is this low level of inflammation really starts to drive a lot of chronic disease. And so we have tied mitochondrial dysfunction-based inflammation to, as I said, cardiovascular disease. It's responsible for muscle loss, gut dysbiosis, metabolic problems, and then certainly cognitive decline. And so from this perspective here, we decided to revise our hypothesis to say, could these maladaptive changes in mitochondrial function also predispose offspring to accelerated aging and an increased predisposition to chronic disease? And so to answer this question, what we did was to put our model, or take our model and adapt it into an aging paradigm. And so mice are actually a very, very good model to study aging because they age somewhere between 10 to 20 times faster than humans do. So you don't have to wait too long to get your experimental outcomes. And so the period that I want to show you here for the rest of our talk is this one right here, right around middle age. So this is a period when we go into our doctor's office and our cholesterol levels are starting to creep up and they start reminding us to perhaps eat a vegetable once in a while. And so this point here, our idea was that if this is kind of the transition point, can we detect an early shift in these inflammatory markers as well as markers of chronic age-related disease? And so that's what we did. We used the 2x2 factorial paradigm, but instead of stopping our experiments during fetal development, We allowed offspring to be born, and then we basically just left them alone and allowed them to age out to their middle age, into their equivalent of being almost their late 40s or their 50s. All right, and so one of the caveats to animal research is going to be housing conditions. And so if you grabbed a human and said, I want to study aging, and then you threw them into solitary confinement, you're probably not going to be studying some very relevant outcomes here, because of course they're going to have a rather harsh existence. And so with this kind of perspective, we implemented some very robust cage enrichment. We had extra nesting material. We got them these huts, as well as a different kind of chew toys. We basically did the very best that we could to make the mice happy as they stayed in their cages, essentially, and aged. So yes, there's a caveat here that they were caged. They're not free animals, but this is kind of the constraints that we had. All right, and so the outcomes that we chose to measure from this, we stole from the gerontology research. So there's really a lot of excellent and very well-established research into aging. And so a number of these can be tied to inflammation outcomes, as well as to something called senescence. And that's basically where cells decide to stop dividing. And so the first one I'm going to show you here is the secretion of senescence-associated beta-gal, and that's the blue stain in here. And so what I'm looking at is in the brain, and what we see from our analysis here is that it doesn't really seem to matter whether it was mom drank, dad drank, or both parents that drank. We see an increase in this senescence and inflammatory marker in the brain, with a slightly higher effect in females than in males. When we switched to the liver, we found that there was an increase in fibrosis. And so what happens here when you have that increased inflammation, you have increased infiltration of these immune cells into your liver, and they start to lay down scar tissue. And when we looked at this particular outcome, what we saw was, again, we had a strong signal from mom drinking and dad drinking, but again, when both parents drank, we had this additive interaction that produced an outcome beyond either parent individually. And then, of course, once you start to get this scar tissue forming, you're going to have an increased predisposition to developing fatty liver disease. And so we stained our tissue sections here for markers of fat accumulation. And again, what we saw, and this is in the male offspring, was that there's a strong signal here from mom and dad, but again, it was beat by the interaction between mom and dad drinking. Okay, so then the last thing I'm going to speak about here is kind of how this memory here, this memory of alcohol exposure, is going to persist and then perhaps increase predisposition to chronic disease. And so what I'm showing you here is a measurement of the ratio of NADH to NAD. And so I promise you I'm not going to get into the chemistry here, because I myself am not qualified to talk about it. But basically the NAD plus is the high energy state, the NADH is the used up state. And so you can tell how good your engine is running by how much of the available energy you have versus the used up energy. And so when we look at these aged mice, what we see here is kind of this declining pattern of the availability of this particular high energy substrate and a higher ratio of the used up energy. And so this really suggests that this mitochondrial dysfunction, or this lower level of energy production, is persisting on into adult life, and that we have this co-occurring inflammation that's going along with it. All right, and so now, because of this emerging interaction that exists between mitochondrial stress, inflammation, and the development of cancer, we became very curious whether or not this inflammatory signals will actually predispose or increase susceptibility of these offspring to developing cancer. And so what we decided to do was to focus in on liver cancer, because that's where we'd seen the strongest signal in our mouse model. And to do this, we used our dual parental exposure paradigm. Again, we split them into two groups. The first group got an injection of saline, while the second group got an injection of this carcinogen right here, diethyl nitrosamine, okay? And so this is basically the carcinogen that used to be found in deli meats. And so this, in this model, in the male offspring, it's going to cause eventual hepatocarcinoma or liver cancer. And so we gave the mice the injection of this particular carcinogen at postnatal day 14, and then we waited until postnatal day 182 to look at the outcomes. And so when we did this, what we saw was that the offspring of alcohol-exposed parents had an increased number of visible tumors, and that the tumors that we were able to identify were larger than what we saw in the control population. And so in essence, you had an increased number or increased propensity to develop cancer, and the tumors seemed to develop faster in this particular model. All right, so to summarize my research program to date, we have found an immediate impact of paternal alcohol exposure on sperm. We see evidence for increased reactive oxygen species in the sperm themselves. And when we use this alcohol-exposed sperm in an in vitro fertilization system, we see a dose-dependent decline in pregnancy outcomes. And so you basically have the more you drink, the less odds of you becoming pregnant. We trace these effects to increased reactive oxygen species production and mitochondrial dysfunction in the placenta, and we can also identify this in the brain. And in the developing face, we've identified growth abnormalities in the formation of the face, as well as the lower aspects of the jaw, and these patterning defects persist in the offspring into adulthood. And then, of course, the stuff that I spoke to you about here today, which is going to be the effects on aging. And so we see increases in age-related outcomes. We can tie that to increased reactive oxygen species in the animals themselves, in their livers, as well as other tissues. And these increased reactive oxygen species in the mitochondrial dysfunction associate with increased susceptibility to adult disease. And so the pathway that we're moving on now is to think of alcohol exposure as a driver of accelerated aging, and to begin looking at interventions that are going to target mitochondria to improve mitochondrial health, and then hopefully push back against the development of these age-related outcomes. And with that, I'd like to acknowledge the people who did the work. The studies of the mitochondrial biology were done by Sanat, who is now doing his postdoc at Penn, and the cancer studies were done by Ali, and she's now doing her postdoc at MD Anderson. And of course, I would like to thank funding. I was supported by the Keck Foundation as well as the NIAAA. And with that, I think I'll just acknowledge kind of what a wonderful group and a pleasure it has been to work with these kids and watch them develop. And I think we're ready to take questions. All right, thank you so much.
Thank you, Dr. Golden.